This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 566, recorded on September 20th, 2019. I'm Vincent Racaniello, and you are listening to the podcast all about viruses. Joining me today from Western Massachusetts, Alan Dove.
Good to be here. And here in Western Mass, it is freaking gorgeous and has been for a week, and it's supposed to be for another week. And this is just glorious, clear blue skies. And right now, 74 Fahrenheit, 23 C, just light winds and beautiful.
It's 24 C and sunny here. Nice day. It's just too cold at night, though. Don't like it.
I like that. It's better for sleeping.
Yeah. Also joining us from Austin, Texas, Rich Condit.
Hi, everybody. We have 92 degrees. That's 33 C. We have had so far today at the official weather recording site in Austin, 52 days, I think it's now, of 100 degrees or warmer.
Ow.
But, you get used to it.
It's a dry heat.
Believe it or not.
Yeah.
And it's not too humid. It's a sunny day. I enjoy the mornings. I even go out in the garden, mess around. So it's all good.
So in a couple of weeks when I'm there, it'll still be warm, right?
It'll still be warm, yeah.
All right.
But by the time you're here in a couple of weeks, with any luck, we'll be down under 90.
It's fine. Still sounds good. Also joining us here in studio, back for a second time, Kiki Warren. Welcome back.
Hello, all. It is, as said before, a nice brisk day in New York, and I love the cold night times here. So much better.
Now, I'm going to move where it's hot all the time.
Texas, it seems like.
Austin will do that for you.
I don't know what your affiliation is. Are you a Columbia University student?
Yeah.
You're still a student?
Forever a student, I think.
Do you have another semester to go?
Well, last semester right now. Last semester at Columbia. And
in the fall. That's what we're in now.
this current semester.
Are you taking classes?
A couple ones, yeah.
Which?
Global Threat of Antimicrobial Resistance.
Wah, wah.
What else?
Biopharmaceutical Regulation and Development.
Wah, wah.
And?
Biochemistry.
Oh, that's good.
Oh, cool.
Biochemistry is important.
Everyone's like, oh, that one's fine. less dark, I guess.
I have to say, the other ones are made and taught to attract students to take them because they're topical and contemporary. But you need to know the fundamentals.
Good professors as well.
You need to know the fundamentals before you understand antimicrobial resistance.
That is why I took your course.
Like how an efflux pump works. Well, I can tell you about antiviral resistance, but not antimicrobial. Someone else.
Which is why I took the class with Mausch.
Yep, Moschwitz. Did he teach you? He. Well, he teaches immunology. So it's not going to tell you about
immunology.
It's not going to tell you about efflux pumps. No, those are not immunology. That's what pumps out the tetracycline and so forth. Anyway, welcome back. And those listening, consider supporting our shows if you like what we do. Microbe.tv/contribute. We'll have more about that later. But just to say, we all do this because we love it. We don't do it for profit. And there are expenses involved, so consider contributing. Now, my latest trip, I was in Sweden in August, which was a lovely place, and I've talked about that. And last week I was in Denmark, which I'd never been to either. What a great country. 5 million people. Any of you ever been to Denmark?
No.
Yep, long time ago.
Lovely, lovely country. Had a good time. Shout out to all my friends that I met there. I went to Aarhus. So I was saying it Aarhus before I left, and then they corrected me. It's Aarhus.
Aarhus.
Aarhus, which is the second largest city in Denmark and has the biggest university, University of Aarhus. And Maddie, a PhD student, invited me to go there when she heard on TWiV that I was going to Denmark. So I have upcoming trips. Let's see. Next year we'll be in Finland. I think it's in June. So if there's anyone there that wants to have a podcast, let me know. And I probably will go back to Sweden at that time, maybe visit Umeå or Gothenburg, where there are strong universities with virology programs. Do some
also in
podcasting.
Stockholm.
I could go back to Stockholm. There's a lot to do there. I never got to see the archipelago, so I'd like to do that. And then I went to Copenhagen for, this is a cool meeting actually, European Society for Clinical Virology, pathogenesis, epidemiology, diagnostics, antivirals, a lot of the stuff that we don't hear about very much at other meetings. So it was really, really cool. So we'll have 2 TWiVs coming from Denmark in the coming months.
Cool.
They're in the can. I also have another Sweden episode in the can, the 3rd one. And,
so in Sweden, you met my friend Sophia Appelberg?
I did, and we read her email last time. You weren't here. But yes, I did meet her, and she was very enthusiastic. Mm-hmm. And I think she got a T-shirt that we threw out.
Good.
So she was very excited. So she works in the lab of Ali Mirazimi, who was on the one of the TWiVs we recorded there, the one I released last weekend. And she does work on Crimean Congo hemorrhagic fever virus.
Cool.
And they're trying to make a vaccine for it. So let's see what's coming up next. Next week I'm going to Reno,
Nevada.
University of Nevada, Reno.
That'll be hot.
You think?
Yeah.
Desert?
Yeah.
Isn't it high desert?
Wouldn't that be cold at night too? You hate that part.
Might be.
Yeah. Well.
Worst
anyway, so that'll be fun. Then, what's after Reno? Oh, then I go to Austin and Galveston with Rich Condit following week. Right, Rich?
Yep. We'll have a good time.
So Rich and I are going to do a TWiV in Austin. Then we're going to drive to Galveston. Road trip.
What do you guys bring on a road trip? What's the one thing you must bring on your road trips?
A car.
What?
It was a question.
No,
is there a Gatorade? Is this a Red Bull scene?
No, we just talk.
The Impossible Burger?
Oh, by the way, Rich, I saw a friend of ours just bought a 1962 or something, early blue convertible Mustang.
Oh, man.
It's like a vintage thing. That's what we wanted to drive last time. When I went to Florida, we were going to drive a yellow Mustang convertible.
Canary yellow convertible Mustang. That's what TWiV road trips ought to be on.
All right.
Hey, it looks like it's going to.
Road trips ought to be like.
Yeah. Looks like it's going to be comfortable in Reno.
Good.
Like we're talking, it's all over the place, but anywhere from mid 50s to mid 70s. High. There's a high here of 81, so you're going to be plenty comfortable.
So then the 15th of October, I'm going to Georgia to do a TWiV at Georgia Tech. Then I'm going to pop over to Georgia State and do a TWiV there.
Are you doing this all as like one big trip
to be
around the country?
No, I'm coming home in between.
Coming home in between. Okay.
I would be in big trouble if I didn't. All right, then a couple of weeks home, and then in November, Rich and I are going to the giant virus meeting in Germany.
With Nels
Zelde. We're going to do a TWiV-TWiVo fusion. And then in December, I'm going to Singapore.
Wow.
For a TWiV at the Nipah virus meeting.
Have you been to Singapore before?
I've never been, so people tell me it's amazing. Anyway, if you're in any of those places and you want to...
My grad school roommate lived in Singapore, he got a research position there, and was there for several years and really, really seemed to like it. Yeah. It was Bill Burkholder. I don't know if you knew him.
Yeah, I know the name for sure. So Linda Coughlin, who was on TWiV, was just there last couple of weeks for a flu meeting in Singapore. And she said it's her 3rd time there, and she said it's great.
Cool.
All right. So that's the travels, the peripatetic podcast or podcaster. We have a follow-up from Sean. Hi, Vincent. Love the podcast. Writing in from sunny San Diego. Just wanted to send in a comment that, to my understanding, using fetal tissue for research has not been outright banned in the country yet. Yeah, yet is the operative word. The NIH is not allowed to directly use fetal tissue, and therefore direct contracts, such as the one they had with UCSF, have ended. Also, future grant funding, and I'm assuming renewals as well, will have additional ethics processes. So right now, labs that have funding are allowed to continue the research. So this refers to a story we did a couple of weeks ago where they implanted human fetal lung tissue into the backs of immunosuppressed mice and made little organs that they could infect with viruses. So here's the story from the scientist. July 26, NIH announced new restrictions on human fetal tissue research. Starting in September, scientists writing grants for experiments involving this kind of work will be required to explain why other tissue types can't be used and where they will get the tissues. This does a pretty good job of doing what the pro-life people want. Makes grant applications a lot more onerous, someone quoted. So that's it. Latest restrictions. Don't make tissue research.
Quite a quote from Carolyn Coyne here at the end.
Oh, yes, Carolyn Coyne. In addition to the detrimental impact this will have on medical research, these new restrictions highlight the unfortunate trend of politicizing medicine and research. We have seen the same trend with climate change and vaccines, says Carolyn Coyne, professor at UPitt. Yes, everything's being politicized, if you haven't noticed.
Oh, yeah.
Even auto emissions.
It's all of society right now.
Well,
Carolyn comes right out at the end here and says, my concerns relate to more general concerns I have regarding our government and elected officials. Do they really want to protect human health, or do they want to be reelected?
Yeah, they want to be reelected. Hell with human
reelected.
health. It's all about reelection. So coming from Sweden and Denmark, it's a lot less of a problem there. People are really nice in those countries. I never had anyone yell at me or tell me I shouldn't be doing something. Never honked their horns. Everything is cool.
Just jaywalked all the time.
No, you don't. You don't jaywalk. Even though they're supposed to stop for you. I didn't trust them because I'm from here, right? Because my host was, you can walk. It's fine. I said, no, I don't trust them.
I just play Frogger out here.
Both countries, they have very small populations. It's much easier to do things with a small population, like get 95% vaccine compliance. But they do have anti-vaxxers in both countries.
Really?
Nevertheless.
Are those native communities, out of curiosity?
I don't know. That's a good question because they do now have increasingly more and more immigrants, right? Refugees and so forth.
Yeah, maybe people also from California going.
No, they're probably endemic. I mean, there's stupidity everywhere.
Unfortunately.
I don't think any culture gets past that.
But they're having their issues in Sweden, especially with lots and lots of immigrants that are changing the landscape of the country, and some people don't like the nationalist movement like we have here, right?
Yeah.
Got to keep everything the same. It's ridiculous. As if.
Well, the difference is, I mean, Sweden was not built with a strong immigrant tradition the way the U.S. was. I mean, the U.S. we're all from someplace else. Sweden, they've got their own history and certain uniformity to it that is now being challenged.
I say, you have to embrace change, right?
Of course, yeah.
Depends the type of change.
Yes, right.
Well, right.
Yeah, that's absolutely right. But they, in fact, in Sweden and Denmark, and maybe Finland, they eradicated polio using inactivated polio vaccine.
Yep.
And Erling Norby pointed this out to me when I was in Sweden. He said, Albert Sabin said it couldn't be done, but we did it here in Sweden.
So is that still the case?
Yeah, they use IPV exclusively, and that's because they could immunize over 96% of the population, no problem. Get immunized, sure. Which arm? Not yet.
Well, and it's. I mean, if you have a wealthy country with a well-functioning healthcare system, then these are the things you can do.
So Sweden, maybe Denmark too. Free healthcare, free college, free medical school. Holy cow! And the
small
schools
populations.
Are good. The schools are good too. Can you imagine going to Columbia for free?
Well, the medical school is maybe becoming free for some people now.
It's true. If you
changing.
Needed to borrow money, it would be free. But if your parents can pay for it, it's not free.
That's definitely not free.
But there, it's all free. It's great. I think it's a great model, but you can't do it with all the people we have, right?
You could, I guess. It'd just be hard to implement. And
I think the bigger issue is you'd be goring too many people's oxes. There's just too much vested interest in the current system here.
All right, we have a snippet in a paper for you today. The snippet comes from a young man I met way back in May in Rotterdam at the European Society for Virology meeting. He said, "Can you come to my poster? This is the first author, Karim Ezat." Or Ezat, sorry. Don't know how to put the accent. And I went to his poster, and it was what's here in the snippet will do. And then in Sweden at the Karolinska, he showed up for a TWiV because he works there. And he said my paper was published. So I said, we'll do it, because I really thought it was a curious observation, as you will see.
It is.
The name of the paper, which is from Nature Communications, the viral protein corona directs viral pathogenesis and amyloid aggregation. And so Karim is the first author, and then we have two co-second authors, Anna-Lena Spetz and Samir Andalusi, and these come from
co-senior authors.
Co-senior author. What did I say?
Second authors.
Co-senior authors.
There are about 20 authors in between Karim but then
yes.
co-second authors.
And they're from Stockholm University, the Karolinska, University of Oxford, the Discovery Institute in La Jolla, Cairo University, the University of Helsinki, University of Eastern Finland, Queen's University Belfast, and Evox Therapeutics in Oxford. Wow, it's an interesting mix.
Yeah, all over the place.
The protein corona. Now,
yeah,
Alan, tell us what a nanoparticle is.
All right. So when I looked at this paper, I was like, wait, the protein is called corona? No, it's a coronavirus. No, it's oh man. Now we've got a whole new corona term in virology,
and don't forget beer. Don't forget beer.
Right. So the nanoparticle people, and these are mostly physics, chemistry type folks, had discovered in making these engineered nanostructures that if you put them in biological fluids, schmutz sticks to them.
Schmutz, that's right.
So proteins that are floating around in blood or in any other biological fluid will stick to nanoparticles, nanostructures, and they form this sort of halo of proteins that typically is referred to as a corona. Right? So It's like the corona of the sun or of a star or something. You've got a protein corona around the nanostructure. And so these folks said, well, viruses are nanostructures.
Yes.
Do viruses get these protein coronas? And as we're going to see, the answer is yes, they do.
So there are two viruses here, respiratory syncytial virus, a respiratory pathogen of very, very young people. Herpes simplex virus type 1.
But not coronavirus.
Not coronavirus. They grow up the viruses, they purify them, and they dip them in various biological fluids.
Now, RSV is an enveloped RNA negative sense virus, and of course, HSV1 is an enveloped DNA virus.
That's right. And they use human plasma. They use juvenile plasma. They use
an important difference there is that the adult human plasma has anti-RSV antibodies in it because people get respiratory syncytial virus all the time.
And the juvenile were negative, right?
Juvenile were negative.
They use human bronchoalveolar lavage fluid, BALF, where they put fluid in your lung and suck it out again. So
that was also RSV positive.
This is not a...
Sounds like a fun procedure.
That really
and BALF is the sound that you make while they're doing it.
Rhesus macaque and fetal bovine serum, which you can just buy and which we use in our cell cultures. All right, so those are the different ones. And they make viruses, they purify them without serum, and they mix them with the fluids, and then they ask what is sticking to the virus particles, and they use mass spectrometry to figure out what proteins are there. You couldn't do this experiment, 15, 20 years ago, I guess. And now you can do it. And they get a lot of protein sticking to these viruses. And
I like the top 10 proteins in the protein coronae of the virus.
Yes, the top 10. It's the top 10.
Top 10 lists.
They have, the interesting thing, I think, is that each virus, the two viruses, the proteins that stick to each one are unique and also dependent on what fluid you're putting the viruses in, right?
Yeah, there's some overlap, like serum albumin shows up repeatedly, but there's a lot of diversity in these.
And the most abundant ones in the corona were not necessarily the most abundant proteins in the fluids.
Right.
Which
Right.
is good, because otherwise you'd say, it's just mass action sticking, right?
Right.
Characteristic viral sticking thing with each fluid. And they looked at these via transmission EM to visualize the corona. I'm thinking of those things up in the sky. What are those?
Yeah.
The northern lights, right?
Right.
Ooh, we saw the northern
That's
lights.
an aurora.
It's an aurora. I'm confusing
corona.
coronas and auroras.
Right.
Corona's
corona,
around a star.
Yeah, that's the halo around a star, around the sun, or it could be part of a flower. Anything crown-like in nature tends to get called a corona.
They also did some cryo-immunoelectron microscopy where they took antibodies against certain... there you go. That's a corona. It's like a solar eclipse thingy, right?
Yeah.
And see what proteins are bound to the virus particles, like surfactant protein A. They could use antibodies and show that. Yes, in fact, these proteins are stuck onto the surface of these virus particles. And they have some nice photographs showing you these corona. These have always been there. We just never looked, right? Who looks at their viruses in the electron microscope? Not very often. So we didn't see them, but they're always there. So does this affect infectivity? So you could infect with and without a corona. And they infect Hep2 cells. In culture, with RSV, there was an effect on viral infectivity. Now, if you use the adult human serum, it reduces infectivity, and that's, of course, because there are antibodies to the virus in there, so that makes perfect sense. But if you use some of these other fluids, some of them actually increase infectivity.
Actually, the RSV plus the human plasma, looking at the top 10 proteins, what I noticed more than anything else is it's got a bunch of complement components on it.
Yeah, they have immune components, right?
There are a few you get complement sticking from the human plasma sticking to the herpes as well. Mm-hmm. But it doesn't show up in any of the other samples. In particular, if I'm not mistaken, the complement doesn't show up in the juvenile human plasma.
Mm-hmm.
Right, which struck me as, interesting. But at any rate, that might account for some of the effects on infectivity.
Yeah, it would be interesting to add some of these individually and see what's doing what, right? I guess that might be what they're doing.
I imagine that would be, yes, the next logical experiment.
Fetal bovine serum and the monkey serum, they got 5 to 6-fold enhancement in infectivity. Also, the BALF enhanced infectivity, but not as much. It's interesting. BALF, remember, is the fluid coming out of the lung.
Coming out of the lungs, yep.
And that's where the virus is going to infect, so maybe there are things there that enhance infectivity.
So I think the if I'm not mistaken, correct me if I misremember this, but the sort of general explanation for the enhancement of infectivity is that the corona had like sticky type proteins in it and things involved in adhesion.
Probably. I think that makes sense, right?
Right. And there's also, one of the things they mentioned in the introduction is that these coronas, when you see them on artificial nanostructures, they can cause reactions because they're raising the concentration of these proteins locally within the corona, and can expose cryptic epitopes and generate toxicities that wouldn't have occurred with just the nanostructures or just the proteins in the fluid.
Mm-hmm.
So you can change the structures of, so some of these proteins that we're looking at in these lists may not be the shape that they are normally. So the virus may actually be altering them, and that may be part of the increase or decrease in infectivity you're looking at.
They also show that the BALF-coated virus particles can activate dendritic cells. So the fluid from the lung can, the dendritic cells would be taking up foreign particles and presenting them. So I don't know, maybe this is some factor that's involved with that. Yes. You're raising your hand. Oh, you're not?
Habit.
Oh, it's habit. I thought you were like, you wanted to ask a question.
No,
right.
I will ask a question.
You don't have to. Go ahead. It's fine.
Do you guys think that the enhanced aggregation of certain particles could help with some sort of targeting against different viral molecules, sort of in the different areas? So depending on what is attracted to them, you could try to tag one of the things that's more likely to go towards or aggregate towards the corona of the virus to try to target or eliminate the virus.
You're talking about an antiviral approach?
Yeah.
Yeah, if these are really important, I mean, it's only a 5 to 6-fold enhancement, at least in cell culture, right? The key is in an animal, what these are doing. But if that were only 5-fold in an animal, then that wouldn't be enough. You'd have to
on the other hand, if there is a reaction that reveals some cryptic epitope on these, you might be able to target that.
Mm-hmm.
The concern I would have with targeting these proteins as they are is that these are proteins that are just normally circulating, and that's probably not stuff you want to go shooting at. You don't want a compound that's going to target complement, because that's going to have side effects. But if you have some epitope on the complement that gets uniquely exposed when it's in a viral corona, then that would be a great target.
Yep. As Rich said, these inhibitory fluids comprise antibodies, complement factors, immunological things. If you do gene ontology, you find immunological related proteins. And then the enhancements are things related to adhesion, anchoring, protein targeting, etc. So it kind of makes sense. All right, so then the second part of the paper, they say, okay, nanoparticles are known to bind amyloidogenic peptides, little peptides that can give rise to amyloid, which are aggregates, tangles of proteins, and characteristic of things like Alzheimer's, the AD, the Allen-Dove disease. Oh, by the way, Alan, I did a paper in Stockholm, and one of the first authors' name was alanine. Was that the paper we did?
Oh.
I didn't get to see the alanine paper, but I would have loved to see the alanine paper.
So, yeah, we did a paper, and I don't know if it was the last one where we did it together. But anyway, the first author was alanine something, so I thought of you.
Oh.
And then the person wrote in.
I would have just scanned the paper.
The person wrote in and said, yeah, people do talk about alanine. Did you get his joke? I would have just scanned the paper.
Yeah.
You do? You know about alanine scanning?
Not in depth. I have heard it passing enough to know that it was a joke.
You just change every amino acid to alanine, right? As you go down the amino acid sequence, because it's such a is this
such a boring amino acid.
Right? I was going to ask Alan, is that no? But you're not boring, right?
I don't know.
No, you wouldn't be on TWiV if you were boring, right?
Exactly.
If you were boring, I wouldn't have taken you as a graduate student. You can tell boring people, right?
Pretty quickly. Yeah. You're great, Alan.
So, and nanoparticles bind amyloidogenic peptides. So do these viruses bind amyloidogenic peptides? And there are a number that you can test, and they add them, and they do, in fact, bind these peptides, and they make fibrous networks. Not every peptide. I think RSV just bound one of the two. HSV also bound an amyloidogenic peptide and accelerated amyloid formation.
These are amyloids that are associated with either Alzheimer's or type 2 diabetes, right? There's an islet amyloid. This was a new one to me. So I guess it's implicated in type 2 diabetes, wiping out islets because of its precipitation.
Some people have actually proposed that Alzheimer's disease should be called type 3 diabetes.
Mm-hmm.
Really?
It's kind of out there, but there are a lot of parallels between the pathogenic processes, and there's some reason to argue that. Anyway, these authors are very, very big fans, from what I can tell, of the HSV causing Alzheimer's hypothesis, which they refer to it as the presumptive role of HSV1 in the pathology of Alzheimer's disease. And my little margin note was very presumptive at this point. But but it is interesting that this you get this viral interaction going on, and this is a potential mechanism that could be involved somewhere.
So if you remember from our previous TWiVs, the idea is that multiple herpes simplex type 1 infections of the brain leads to deposition of amyloid being a antiviral response to kind of encapsulate the virus, but it can also have detrimental consequences. And here, you just mix virus with the peptide and it will make fibrils.
So the peptide that they focus on for the RSV experiments is the islet amyloid peptide. And for HSV, they focus more on the Alzheimer's peptides. And in both cases, they associate, and the various treatments seem to dampen that effect. So if you pre-treat RSV with fetal bovine serum, it sort of tamps that down. Same with, same with HSV.
So the bottom line is, before you get infected, you should coat your viruses with fetal bovine serum.
Yes.
Well, could you inoculate in that way? I don't know.
No, you can't, because you don't have a choice when you get infected, right?
I mean, if you did it at an infant stage, maybe.
You don't want to inject people with bovine serum.
No, we don't want to. We just vaccinate. That'll be the key. And then the last experiment is to say what happens in mice. Now, this is an experiment that we've talked about as being done in other papers as well. They take these transgenic mice, which have they produce human proteins, and they have 5 changes in them that have been linked to Alzheimer's disease. So they're considered to be a model for Alzheimer's development. And they infect them with herpes simplex type 1, and they get amyloid deposition in the brain, in the hippocampus, the cortex of these mice. But it just means that when you put the virus into these animals, you get amyloid, right? I guess they see specifically this particular peptide, alpha A beta 42, which is the same peptide that they found stuck to the virus. That accumulates in mice. So I guess that's the connection between the two experiments.
Right.
And that, by the way, I think it's relevant, was an intracranial injection.
Yes.
So they're shooting HSV right into the brain.
That's right.
Which is not the normal route of infection for those.
Right.
Oh, really?
And they're not doing, they're not messing with this is kind of separate in my mind from the corona experiments. There's a correlation here between the fact that HSV will aggregate amyloid and the fact that you can get amyloid deposition if you shoot HSV into the brain. But there's a pretty long distance between those 2 points, A and B.
Yeah.
And also other papers have explored this herpes connection more thoroughly, right? We did one from an Italian group that characterized this mouse model extensively with herpes simplex. In fact, they did reactivations, right? They established a latent infection and reactivated, which would be more a normal route. So, but I think what they wanted to do here is make a connection between the A beta 40 peptide or 42 and what happens in a mouse. So
they say several recent studies have suggested that A beta 42 is an antimicrobial peptide that aggregates to sequester pathogens.
Right.
And they say that theirs is an alternative but not mutually exclusive explanation. I don't see how it's, to me, this result and the result with the corona or the virus being able to aggregate amyloid is completely consistent with the notion of the peptide having antimicrobial activities. It all fits together to me.
So that's this paper. It's kind of interesting. I think the cool part is that a lot of proteins can stick to the virus and make a corona. And now we know that we have known before that proteins can stick to virus particles, of course. But here we have a whole proteome stuck to the particles, not just one.
And it differs from one particle to another.
Right. One virus type to another.
One virus to another.
Yeah.
Yeah.
And so, the next part, I think, is to dig deeper and see which of these is doing what, right? Which proteins is doing what for which virus, and what about other viruses? It probably is a universal thing, I would guess. So look for more on this. I thought
it was interesting to see this on some unenveloped viruses.
That's a good point, yes.
I forget where it was in the paper. I'm looking for it, and I can't find the data. Maybe it was just in the text. But if I recall correctly, they did an experiment where they coated RSV with the various coronas and then tested to see if there was reaction with an anti-RSV antibody, this monoclonal that is used for treatment of RSV. And they found that the antibody still bound and neutralized well. So I think that's important. The corona does not occlude the binding of a high affinity antibody.
Yep. I think looking at a non-envelope virus would be very interesting, and I would predict you would get proteins sticking to them as well. Yeah,
and different.
Sure. Different. Yeah.
Yeah. I mean, there's schmutz everywhere, and it sticks to everything. We shouldn't be surprised. I thought it was kind of an interesting finding. There you go. It is. And we'll see what happens next. Okay.
I do wish it wasn't called a corona, but I don't think we can do anything about that.
Is this the first paper about it?
It's the first time they've shown an array of proteins. I mean, we know single proteins that stick to virus particles that have been studied before. Single proteins, but not a whole collection of them. And that's because they did mass spec. You just have to look, and you will find, right?
It's all of science.
Yeah. All right, the next one is a bit different. And this immediately caught my eye. Nature Microbiology, a proofreading-impaired herpesvirus generates populations with quasispecies-like structure. Trimpert, Grunke, Dunek, Kunek, Eschke, He, McMahon, Osterreider. This comes from the Freie Universität Berlin and the BAM, Federal Institute for Materials Research and Testing, both in Berlin. And so the word quasi-species got me because we've been talking about quasi-species.
And apparently you're still allowed to say it.
You see, there it is.
The title of a Nature Microbiology paper. So,
quasi-species-like.
Yes.
Yes.
It's a soft quasi-species.
It's a quasi-species.
we
quasi-squared.
We got some emails about this. And I have to say that so the first person to really apply this word to viruses was Esteban Domingo, who a long time ago, while studying a bacteriophage, 1978, he published a paper called Nucleotide Sequence Heterogeneity of an RNA Phage Population. And we weren't able to do sequencing back then, but he could do RNA T1 fingerprinting, where you chop up the RNA into pieces and then you run them in 2 dimensions on a gel, and the pattern is kind of diagnostic of the sequence. It's an amazing technique. It's great in the paper. They say, although we are not able to sequence these RNAs, we can look at their composition. And he said, let's call this a quasi-species, because he said we have a lot of variation in the sequence of this phage. One thus just arrives from his paper. One thus arrives at the concept of a phage genome, which is statistically defined but individually indeterminate, insofar as most of the individuals differ in a few positions from the average sequence. On the basis of theoretical considerations, Manfred Eigen has predicted this situation and coined the term quasi-species to describe such a population. All right, so that's first time it's used for a virus. So yesterday I emailed Esteban in Madrid, which is where he is.
Wow.
And he wrote, I'm on vacation, but I will put together a response in the next few days. Thank you for your note. I am well aware of the controversies you mentioned, but there are many arguments to support the use of the term quasi-species in virology. Years ago, I was quite unhappy about the objections, but now I do not worry anymore. And I'm firmly convinced the term conveys the major features of viral population dynamics.
Finishing your quote from this paper, heterogeneous populations of this kind should be capable of rapid adaptation to a changing environment, not only because of the high mutation rate, but also because of the presence of a large proportion of variants. I think this is downright prescient.
Yes, absolutely. It is.
And I note that the paper comes from, he must have been a student or a postdoc with Charlie Weissman.
Yeah, he was doing a postdoc in Switzerland, yeah.
This takes me way back. This gives me chills. This is great stuff.
Just before you were born, 1978.
Yeah.
Not you, but Kiki, right?
Over 15 years before I was born.
Wow. Yeah. I remember this as a grad student. It came out, and I didn't know what it meant, but many years later.
But it was very exciting.
So anyway,
sure when this came out, I wouldn't have known what it meant.
So in this paper, RNA viruses form these quasi-species because they have error-prone polymerases. All polymerases are error-prone, but the RNA polymerases do not correct errors. The DNA virus polymerases can, so they have a far lower mutation frequency. They probably also have a quasi-species, but it's much less extensive. And Kiki asked me before, what's the cutoff? And I don't know. I don't know what the cutoff is. I just don't know. But interestingly, single-stranded DNA viruses have a higher mutation rate than double-stranded DNA viruses. And we mentioned that briefly at our TWiV in Rutgers this summer. Siobhan Duffy works on that, and they don't know the reason for that, but that's very interesting.
I wonder if that has anything to do with the absence of a complementary strand.
I was just going to say that. The complementary strand basically is a spare copy, right?
Yeah, sure. She didn't bring that up. She mentioned other things like nucleotide pools and so forth. But
I mean,
I know
the single-stranded phages always go through a double-stranded replicative intermediate. But if you spend any time as a single strand and you're exposed to some sort of environmental insult that could change a nucleotide sequence, that would be a replication.
Idea what it was supposed to be.
Yeah, right.
So in this paper, they take a herpesvirus with a double-stranded DNA genome, and they trash the error correction machinery of the polymerase and ask what happens. Okay?
I want to talk about that for just a second. Point out that the proofreading function on DNA polymerases in general, including herpes, is contained in a separate domain of the protein.
Same protein, right?
Same protein, but a separate domain. In fact, sufficiently separate so that in the E. coli DNA polymerase, you can lightly treat it with a protease and clip that domain off because there's a hinge region that's exposed. So this is really an accessory function, like somewhere along the line, DNA replication said, "Oops, we need to do something about this," and added a domain.
And this is an exonuclease. It'll chew back the DNA and then put in the correct one to correct the error. So it's a backspace.
Chew back, and give the polymerase a second chance. Say, no, you got that wrong. Try again.
Chewbacca. Did you hear him say lightly digest? Nobody uses such terms anymore. Everybody's blunt, hitting everything. Lightly digest. This is why I wanted Rich on this episode, because he knows all about this
stuff. It's all the finesse.
Yeah.
So that's the overall thing. Trash the exonuclease, the error-correcting part of the polymerase, and see what happens. And they use Marek's disease herpesvirus.
Which is a chicken
because then they could infect chickens with what they get. Because if you have a herpes simplex, and what are you going to do? Infect mice?
Not the correct model. Yeah, this is a natural virus of chickens. So then you actually have an animal model.
And they tried this experiment with simplex and it didn't work, or someone else did.
Well, there was that too.
They say, in contrast to herpes, where they trashed the exo and the virus was completely non-infectious, it works here. So they make changes so the thing that's cool about Marex, very low sequence divergence. Field virus isolates are more conserved than in other herpes viruses. So if you get one from a chicken here and you go to California and you get one, they're very, very conserved. Not a lot of sequence diversity.
The chicken tastes the same everywhere.
Tastes like chicken. That's what they say about frog legs, right?
That's what they say about lobster too. Everything that
lobster
tastes
doesn't
okay
taste like chicken. No way.
If you put it in enough butter, lobster tastes like chicken.
No, I can still tell it's lobster.
So they introduced changes in the exonuclease domain of the polymerase. They have a number of amino acid changes in different domains of this exonuclease, and they produce the proteins and show by in vitro assays that they get rid of the enzyme assay. Very nice, right? This
is a little esoteric here, but I really got off on these mutations in the exo-1 domain because they know from the structure that they can identify amino acids that are important in coordinating, that is, binding a magnesium ion that's required for the catalysis.
Yep.
And so they can change those amino acids to boring old alanine, okay,
yep.
and predict that enzyme will no longer bind magnesium, and that ought to kill it like totally, and it does. Sure. I just thought that was cool.
That's great. It's totally great. It's exactly what you should do.
It's elegant.
It's very elegant. They have two sets of changes. One set completely trashes the exonuclease activity, and the others leave a little bit of residual activity, 20 and 85%, which is a lot. Right. 85% is quite a bit. Okay, so those are the changes they put in the exonuclease. By the way, I have a little difficulty going through this paper. It's a bit confusing, so hopefully between the four of us,
we'll get it straight.
Get it straightened
By
out.
the way, I don't know if we ever did the big picture here, but the idea is that if you get rid of the error correction, you should generate a virus that makes more mistakes. And the question is, what effect is that? Do you get a quasi-species of DNA viruses, or what effect is it?
Exactly. So they put these changes into the viral DNA, and then they transfect the DNA into cells, cultured chicken embryo cells, and they say, they look like wild-type virus. We put these changes in, and we see cytopathic effect. However, if they then took the virus and passed it, infected another set of cells, that's one pass, infect another set, that's what we mean by passage. I've learned that not everyone understands what passage is, so that you take the yield from cells, you infect another cell, on and on. It doesn't grow well on these passages.
After the fifth time, it's almost completely gone.
Yeah, it declines passage to passage, so it's the errors are presumably accumulating, and it just gets worse and worse, and things fall apart.
It's on Muller's ratchet.
Muller's ratchet. That's right. It's right.
The decline is consistent with what sort of mutation you put in, because some of these mutations are less deleterious on the exonuclease activity than others, and the ones that are more deleterious have the effect of basically killing the virus completely after
they decline much faster, right? In proportion to the amount of the amount to which you've damaged the exonuclease, you see this fall off.
All right. So the next thing they do, they want to understand what's going on in some detail. So they isolate individual genomes from these virus stocks that are produced by transfection. So that first transfection, they get some virus. They clone out individual DNAs. They clone them as basically plasmids in E. coli. So you get a single clone, and then they can sequence them and see what's going on and get an idea of the diversity. So you can see we have more mutations or what. So they do high throughput or next generation sequencing, and they say we see more mutations with the lower exonuclease activity.
So I want to give a little more introduction to that, because if I understood this correctly, one thing we didn't talk about is that if you actually continue to passage past, that when the virus dies out is by passage somewhere between 3 and 5.
Right.
But if you harvest the whole thing and really try and force it past that, push past that, harvest the whole culture and stick it on another culture of cells, you can, after 9 or 10 passages in basically all of these, see some virus coming back. So that presumably is some strange assortment of viruses. It could even be revertants, I suppose, and a few of them are actually.
Well, and they get some insight on that with the larger scale transfections, right?
Yes,
that's next.
And so, but if I'm not mistaken, these sequences that they're looking at are coming out of these later passage viruses that are recovering.
I'm not sure.
I'm looking at figure 2.
Yeah.
Viral DNA extracted from virus-infected cells at passages 12, 14.
Right, but
60.
I think those are from the larger transfections.
Yeah, the large population size.
Okay.
Yeah.
But,
So that we should probably just go into that at this point. If they take a larger inoculum and transfect and get a bigger initial population, then they can keep passaging it, and it leads to a stable population, and they go in and they sequence that population. And it turns out that a subset have reverted so that they've got a functioning exonuclease. They either reverted the mutation or they came up with a workaround, so they are doing proofreading. But that's only a subset of the population. The rest of the population is still hypermutating.
Right. Okay.
So it seems like you get this pool that eventually figures out proofreading again, gets that going, and that's able to maintain the larger population of hypermutating viruses, which gives you this quasi-species-like structure.
So these passages are all done under conditions where we're having multiply infected cells, right? So the guys with the corrected proofreading function are healthier viruses and can serve a sort of a helper function.
I think so. I think that's
probably
right?
Yes.
Yeah.
But you still have a lot of hypermutating viruses around, so you get a lot of hypermutation,
and you get a diverse population. Now, before they did that, they picked individual genomes from these early passages, right? And they sequenced them, and those exomutants that have no activity, they had very high error frequencies, 100-fold higher than wild-type virus, and these populations went away within 5 passages. So they can initially infect without an exonuclease, but they make so many mistakes that within 5 passages they're gone. 100-fold higher mutation frequencies than wild type. The other exo change, where you have some residual activity, it can be passed for at least 20 passages, but they see there a mutation frequency about 3-fold higher compared to wild type. And again, you do this by pulling out individual genomes and sequencing them. They do a lot. They do a lot of the sequences. So when you have no exoactivity, the virus cannot be passaged at low MOI. You get a lot of mutations, 100-fold higher mutation frequency. Virus goes away within 5 passages. If you have 20 or 80% exoactivity, you can go further, but you still get higher mutation frequencies. Okay, so the XO1 mutants, they don't grow, so you can't put them in chickens. But the XO3 mutants that have some residual activity, they put them in chickens. Okay? And so
now going into this, you would say that those would be, attenuated or more virulent?
Yeah, I'd say they would be attenuated.
I would too.
That's my bet.
That would be my bet. But so they have 2 different mutants. They have one with 20% residual activity and one with 85% residual activity. And they're both attenuated. But the one with 20% is Y547S, Y567F. That's the one with 85% residual activity. That is attenuated, but it can infect the chickens. Y547S, which has 20%, causes disease just like the parental virus, And of the other one with the lower residual activity, 1 out of 25 chickens get even more severe disease. Okay, so that's weird that happens. So these viruses are attenuated, but a rare number of chickens get more severe disease. Then they do these transfections that we already mentioned, large amounts of cloned DNA, where you can get many, many genomes getting into many cells, and then this population can be maintained for longer passages. And I think, as we've said, that's probably because there are some reversions in there to wild type that are providing helper functions for the other genomes to replicate. And so from these kinds of passages, they pulled out 21 clonal virus genomes. They sequenced them, and they found very high, unexpectedly high genetic diversity. About 100 mutations per genome, but they say not a single clone was represented more than once. So every one of those that they pulled out was different. So basically, you see lots of mutations in these genomes, much more than the wild-type virus have. And there is some, as Rich suggested, there's some mutation. About 40% of the population in these passages reverts at the exo mutations. So they're making wild-type exo, which probably allows the viruses to multiply, but the fraction still has no exo, and that's generating all the mutations. Are we good so far?
Yeah.
I think so.
So the initial mutation frequency, even of these exos with some residual activity, is still too high to be tolerated. So you get some reversion, which allows you to carry this population. It's still a more diverse population than wild-type virus. Okay. So now the next experiment they do, they take these 21 cloned DNAs that they had made, they transfect them into cells. Only 14 of them kill the cell. So a lot of those others have defective genomes that can't replicate. And that's where they think that the replication competent members of the population are maintaining the others, but in those where there's no cytopathic effect, there must not be enough replication competent viruses. All right, now they do a big chicken experiment. Question here is, how important is genetic diversity for virulence? So they have 5 groups of chickens. First group, they use the Y567F polymerase mutant after one passage. To remind you, Y567F has 85% of the exoactivity of wild type.
Right.
Passage 1. There, you can get virus out, but presumably already its sequence is diversified. That's one. So they give some chickens that virus. A second batch of chickens, they give a population, a quasispecies-like population from that mutant after 12 passages. Remember, they were able to get 12 passages from that particular mutant because it has more activity. Number 3, a single clone from passage 16 of that virus. Number 4, a combination of 14 independent clones from passages 12 through 16. And 5, the parental DNA without any changes. And making this more complicated.
What would you expect about virulence? After this thing's been passaged repeatedly, 567F has been passaged repeatedly, I kind of expected the virulence to go down.
Mm-hmm.
Right? Because that's a way to attenuate a virus.
So they find that the population of viruses, that's this 14 mixture, has increased virulence compared to the parental virus. And it had a weird disease phenotype. You got paralysis and death of young chickens, which is typical of virulent strains. Single clone was attenuated as the original mutant. So that passage 1 mutant, that was attenuated. The mixture of clones from passage 12 to 16, which they say represent a more restricted sequence space, is an intermediate virulence. So basically, the collection of viruses with the highest genetic diversity is the most virulent.
Right.
Which I would sort of guess, just because diversity has the most that a selective pressure can choose from.
Yeah. Yeah.
In retrospect, as I was reading this, I was like, oh, yeah, that is kind of unexpected. Or is it? Because the virus has explored a bigger sequence space, and so you'd expect some kind of virulent version of it to grow out if that's going to be successful.
The key here was that they figured out a condition where they could do some passages and mix isolates from each passage, and that gave the population enough genetic diversity so that it had an increased virulence, right? Because if they only did passage 1, that's not enough diversity, apparently, to give you increased virulence.
Right.
Are you okay there, Dr. Condit?
I'm good. I'm good.
So they took these chickens and they did some sequencing of tissues, and they confirmed that the ones that that had virulence, that had high disease after inoculation, they have a more diverse population of viruses compared to, say, wild-type virus. And they can do this, again, by high-throughput sequencing. You could sequence lots and lots you can do many, many coverages of the genomes and get a good estimation for the mutation frequency. So that's the bottom line. You can make a DNA virus with impaired proofreading, and it can generate a diverse population, which looks like a quasi-species of RNA viruses. And, for RNA viruses, we say the diversity of the population allows them to adapt to new environments. So here we put a diverse population in chickens, and the viruses are like, whoa, look at this new house. Let's trash it.
Let's trash it.
And
Project X and chickens.
It is the diversity of the population that allows it to do that.
So there's one of the cool things I like about this is that they take virus from the chickens that were infected with the mixture of clones, and they sequence a bunch of those and determine that there has been a bunch of recombination. They pull out viruses that look like recombinants.
Right.
And, this does not surprise me, but I always like to see it. There's, some of these have over a dozen crossovers. I mean, just crazy mixtures of different recombinant viruses. That's sort of in-your-face demonstration that recombination during infection of an animal can be rampant at a high enough multiplicity. So the other thing I want that's on my mind is that, to me, one of the things this paper says is that fundamentally wiping out the exo function is not good. Okay, the viruses don't like that.
That's right.
All right, a huge fitness cost. But if you manipulate it in the laboratory in a way to sort of help it through some of the initial stages, you can come up with this weird stuff.
That's right.
My guess is that if you put that out in nature, even the quasi-species that you generate, if you put it out in nature and passaged it in for a period of time, you'd probably wind up with viruses that reacquired the exonuclease function and were a stable population of viruses,
or went extinct,
or went extinct. Yeah,
I would be interested in having them identify the mutations that gave rise to higher virulence. Right?
Absolutely,
that could be very interesting.
Absolutely,
and that could be informative as with respect to what makes the virus cause more disease.
So the other thing is running through my mind constantly in this paper is the von Magnus effect. Okay, I think we've talked about this before, but this is an old experiment where you do serial undiluted passage of virus. And what you find is that at high multiplicity, where you have a lot of replication competent virus around, enough to infect every cell at least once, but other viruses around as well, you create an environment where viruses that could not replicate on their own are helped by the viable viruses. And in some circumstances, those non-viable viruses can actually compete with the competent viruses to the point where the replication of the competent viruses is suppressed over time. and you get mostly these defective viruses. And once those accumulate to a high concentration, they can no longer, on serial passage, support the replication by itself. So you keep passaging and you don't get a productive infection. They go away, except for a few cells that still have the wild type viruses. So those guys come blazing back, and then you go through the same thing again. So with serial passage, the titer goes up and down in a fashion that coordinates with the accumulation and the disappearance of these defective viruses. And I always wonder with experiments like this, how that relates to a natural situation. What's the multiplicity of infection that a human experiences with herpes virus? Okay. I don't know the answer to that, but my guess is it's pretty low. So that I imagine that if a quasi-species were released into nature, okay, you would pretty quickly select out the replication competent viruses, and these other guys would go away.
Yeah, they actually bring that up. They say whether virulent mutated DNA viruses can hold a selective advantage and be stably maintained over the course of multiple infection transmission cycles remains to be determined. But high virus population diversity at transmission sites suggests that these dynamics could be more than transient. So that's a good question. Although for RNA viruses, now people, as you know, are really into studying defective viruses. It looks like they are generated in vivo. So the multiplicity, at least local multiplicity, must be high enough to do that.
Yeah, local multiplicity might, there's probably a couple of ways of looking at multiplicity. One is, what's the multiplicity in the initial encounter of a host of the virus? And then as the infection progresses, what happens? I think HIV is probably a pretty good example of this, because there's in a way, in terms of the evolution of the virus, a bottleneck at the initial infection of an individual where you have to have a competent virus at probably a fairly low multiplicity infect a cell. And then once the infection gets rolling, you have an opportunity for higher local concentrations of virus, and there is an opportunity for more sort of help and diversity.
They also mention in the discussion, we refer to highly diverse virus populations as quasi-species, but recognize the controversy over the use of this term. So, there you go. And one more thing I want to bring up. This is very interesting, and goes back to the TWiV this summer with Craig Cameron at ASM Microbe. They say, recently, the view that RNA virus diversity per se is a selective advantage was challenged because the anti-mutator phenotype used in a hallmark study to address quasi-species theory exhibited a growth deficit caused by reduced viral RNA polymerase processivity. Okay, what does that mean? So the emerging view has been that RNA virus diversity is actually selected. You select for polymerases that make mistakes because diversity is good for evolution.
Depending on the species.
Now, that idea came from the study of a poliovirus mutant with a single amino acid change in the RNA polymerase, which makes it make fewer errors. And the original observation was that this polymerase, a virus containing that polymerase, which makes fewer errors, replicates to similar levels as wild-type virus, but cannot compete against a wild-type virus. It loses. So diversity is selected for. If you have a better polymerase that makes fewer errors, it's not going to win anyway. Turns out that polymerase actually has a processivity defect. It falls off a lot as it's copying the RNA. So it does have a defect. And those authors who did that study, they say the key, what is selected for, is replication speed, not making errors. The errors comes as a side effect of having to replicate quickly. So the polymerase has to charge down the RNA, make copies really fast, and in so doing, it makes mistakes. But the mistakes are not what matter. It's the speed. And you may say, why? Well, the idea is that these viruses kill the cells really quickly. So they need to make progeny very, very quickly before the cell dies, and therefore the polymerase has to work quickly, and it makes a lot of mistakes. Now, so that's what one paper has concluded with poliovirus. I would say it's too early to say it's speed and not error frequency that's selected, because there are lots of other viruses, RNA viruses out there that are different and that don't kill cells even, right? And so there would be no selection for replicating fast, yet they do have a higher error frequency. So we'll see where that's going. The reason I'm bringing this up is they say we can't resolve this issue with RNA viruses, but here we have a system where we reduced replication fidelity, but we didn't increase processivity of the polymerase. That's something they looked at earlier. In other words, they've decoupled error-prone DNA replication from processivity so they can study the actual advantage of making mistakes decoupled from polymerase speed or processivity. Right. So they have a good system to study that. And I think that's really cool. This is kind of a little bit of inside baseball here, but I'm very excited by that kind of thing because in the class that I taught you, Kiki, I said error polymerases are selected for. Okay, I say that every year, and now there's a challenge to that, and it's got to be sorted out. But it takes more than one paper to reverse that.
And they thought that it wasn't selected for in that paper?
And they say, no, it's not the error frequency, it's the speed that's selected for. And the error making is a consequence of having to go fast.
But they didn't really prove that. All they proved was that the previous work had a factor that wasn't taken into account.
Yes. So
and
they said
one
does
this. Not exclude the other. They're not mutually
right.
exclusive.
Well, in the paper, they actually show that this altered polymerase has reduced processivity. And they say, aha, what you did was slow it down, and therefore it makes fewer mistakes. And so that's what you're selecting for, speed, not error.
But you could do both. So speed might be the top priority, but for cells that don't die, as you said.
Maybe
not.
I mean, I think you need to look at more viruses and polymerases. It just happens to be that the polio is the best developed, and we have this mutant, but I like this kind of back and forth. I think that's the best part of science, right? Figuring out what's going on. So we'll be back. Anyway, Craig Cameron talked about this in the TWiV this summer. He said, because I said this anti-mutator virus, it replicates fine. He said, it depends how you look at it. And if you look at processivity, it's not wild type. So anyway, it's a cool paper. I really like quasi-species. And we will be talking about it more in the future.
Yeah.
I'm a fan of the term quasi-species. Are you guys not?
I am. I like it very much. No, I like it. I just go to talks now where people trash it. They say, you can't use quasi-species.
What is the reason for not using quasi-species? I know we went over this a little bit before the podcast started, but for the audience, why don't people like quasi-species?
Well, Eigen originally coined it not for viruses, for large populations like, humans or other animals around the world. And so applying it to a virus, a more restricted population of a virus isn't correct. We talked about this a couple of episodes ago. Let me see if I can find the description. Oh, this is a good, here we go. This is actually a listener sent in this wonderful email, which I forwarded to Esteban Domingo. Here we go. The term quasi-species was introduced by Eigen in the 70s to describe early evolution of life on Earth and then later expanded to viruses. However, originally the term was specific. It was defined in mathematical terms and population genetics models used to describe a situation when a collection of closely related clones has a different biological behavior than any single clone. It was argued that in such cases, in evolutionary terms, the quasi-species serves as the unit of selection, not individual clones. Later, virologists started using the term quasi-species to refer to any collection of closely related viruses, especially the rapidly mutating RNA viruses. And so the old guard, the ones who originally defined it, were not happy, and they said, and there are plenty of articles out there that said we're using the term inappropriately. But I think it's okay because what we mean is a very diverse population.
I don't like that the nanotech folks named something corona.
I was about to say that.
Yeah, it's not your corona, it's my corona.
Exactly.
But given the microbiology definition of quasi-species, would it then be allowed and acceptable?
Well, no, so see, Eigen didn't define it in microbiological terms. It's just a population term. And the virologist then said, let's use this term which describes diversity of a population to talk about viruses, which isn't theoretically correct, but I think it's okay because it gives you a very quick summary of what you're looking at. You're looking at a very diverse population, not a single species, even though, as I said before, viruses shouldn't be considered species anyway, but
right.
I think it's fine. So anyway, Domingo will write to us and we'll read it and continue this discussion. I think it's cool.
So I'm looking up Manfred Eigen here, just as an aside, and there's some quotes that are really amusing.
Oh, please share.
He says, a theory has only the alternative of being right or wrong. A model has a third possibility. It may be right, but irrelevant.
That's good.
Another one. In theory, there is no difference between theory and practice, but in practice, there is.
There is.
It's almost like a Yogi Berra quote.
Yes.
Yesterday or the other day on Immune, Cindy Leifer quoted someone, I can't remember, saying scientists would rather share each other's toothbrushes than be shown to be wrong by someone else, something like that. Have you ever heard of that? I've never heard that before.
No, that's a new one.
But it's good, right? Okay. We have a couple
transplant.
Let's do a couple of email.
Woo woo.
All right. So Anthony sends us a link to an article, BBC.com, which says that Pinterest will direct vaccine searches to health sites. Well, that's good.
Yeah.
And the response to that I saw on social media was, for people going to Pinterest for health questions, this might not be the best audience.
It's Facebook that should really change, right? Because they display, when you Google
I think it's
vaccine,
Google. I think if I ever
out.
am sick, and I do know how to read medical journals, but I will first go to Google whatever the symptoms are. Like small red spots on my dog's stomach sometimes, or something of that sort. It's always the first go-to, and there's that little side thing on Google. I've never gone to Pinterest for medical advice. But
my impression is that from this article is that this is a growing trend in the social media.
I hope so.
Trying to suppress misinformation by
trying to fix the problem they created. Yeah. Yes.
But how curious would it be if eventually, say, Instagram took on this whole practice of helping to direct people towards a diagnostic position? Because they're having images of you. We can already tell a lot of diagnostic information from images, say of skin or dermatology things. So say you took a selfie and your Instagram has like a little bubble in the top right corner with an info, and you click that and it's like, by the way, you might want to look into that spot on your forehead. Looks like possibly a pre-cancer. Get it checked out. Here's a link to Pinterest.
Alan, could you take the next one?
Sure. Vissam writes, I'm guessing that's the pronunciation. Hello, TWiV professors. I'm an infectious disease physician at the University of Kansas Medical Center who has been enjoying your Twix podcasts for a few years now while commuting, and I definitely like the journal club type analysis, similar to what we do for medical articles with our fellows. One of my main interests clinically is virology and HIV, so I'd like your input on 3 questions related to that. First, do we know if New World monkeys also harbor simian immunodeficiency virus, let's say those in the Amazon jungle? Second, do gorillas get sick with their own SIV gorilla? Do they develop a simian AIDS like chimpanzees with simian chimpanzee virus? And third, about HIV mechanism of entry into cells. Is it a clathrin-mediated endocytosis process, like it says on Viral Zone, or just fusion at the surface membrane like every textbook illustration shows, including the Viral Zone HIV cycle illustration? Thanks for all you do teaching the public about science. Hopefully we'll see an HIV vaccine in the coming few years. It's a sunny day in Kansas City with clear blue sky. Temperatures 29°C. The high will be 37°C. Humidity 63%. Wind south-southwest at 13 miles per hour. Visibility 10 miles. UV index 3. Air quality good. And I will be off to Mexico City tomorrow for the IAS meeting, where apparently there's a rainy season.
Nice.
Vissam is an associate professor of medicine at University of Kansas Medical Center. And then I guess, Vincent, you asked Theodora.
I did. Theodora Hatsuano, who works on these viruses, right?
We had on the show.
Right. No lentivirus infections in New World primates.
Right.
The observations that SIV-CPZ causes disease in chimps was made in the Gombe sanctuary that has a few groups of chimps that are used to human presence. This allows animal identification, sampling, and close observation than what is possible in the wild. Gorilla populations are smaller. I am not certain whether an equivalent sanctuary exists. It's also unclear what the prevalence of SIV gore is, making such studies difficult.
So you have to get the gorilla in the room.
And finally, HIV fusion occurs at the plasma membrane, does not require clathrin. So cancel the viral zone thing.
So viral zone text is incorrect, but image is correct, I guess.
Right.
Rich, can you take the next?
Okay, Richard writes, this is with respect to the Norby TWiV. Many thanks for another interesting talk. One quote attributed to Rosalind Franklin when she moved to Birkbeck was she went, quote, from the palace of kings to the slums of Birkbeck, end quote. But she was happy working with J.D. Burnell and Aaron Klug. As an aside, the Senior British Science Award is the Copley Medal and has gone to such august scientists as Ben Franklin in 1753, Charles Darwin in 1864, and Einstein in 1925. But it has never gone to one woman, Dorothy Hodgkin, which feels wrong given the contributions of Marie Curie, Esther. It has only ever gone to one woman, Dorothy Hodgkin, which feels wrong given the contributions of Marie Curie, Esther Lederberg, and Barbara McClintock.
I didn't realize that Klug had worked with Franklin. Of course, Franklin was at Cambridge, where Watson and Crick were working.
Yep.
So that's what she meant, going from the Palace of Kings to the slums of Birkbeck.
And by the way, there's a lineage of crystallographers, and Dorothy Hodgkin is sort of a grandmother of the field. Rosalind Franklin worked with her. Several other prominent crystallographers worked with Dorothy Hodgkin, who actually ended up having to teach at the women's college because she was female, and the guys didn't want her in their chemistry department. And as a result, she ended up recruiting a lot of very smart women to crystallography. And so some of the most prominent crystallographers in history have been female as a result of the legacy of Dorothy Hodgkin.
That's a good legacy.
It is, yes. And it's almost unique among hard science fields in that respect, is that you've got, it's not a high proportion of women in the field, but there's a high proportion of very prominent women in crystallography as a result.
Interesting. Ken writes, all these episode titles are very clever, but they do nothing to help me find a topic I'm interested in listening to again.
That's what the show notes are for.
I would say there is no way we could make a title that encompassed everything we talked about on a show, so we might as well make it whimsical, right?
Yeah, we could just say amalgamation every time, but I don't think it'd be as much of a draw.
Most podcasts have clever titles, right?
Yeah.
Sure.
Yeah, it's part of the fun of it.
So, sorry, Ken, just Google it.
Also, you get to listen to find out. Isn't that part of the fun?
I guess.
It's a journey. We all take it together.
And there's always, and the page for the episode, if you do Google this or if you search on the TWiV site, there's always additional text on the page saying what was covered on the episode.
Yep. So Justin sends a link to an article in Science. UC faculty members quit Cell Press editorial boards over impasse with publishers. And Justin wants to know, any idea how much of a big deal this is? How much pressure this applies or doesn't? And I'm sure Alan has an opinion on this.
This is this is definitely something that's coming to a head, I think. And Cell being part of Elsevier is a major target. And the University of California, obviously being huge and prominent, they're clashing on fundamental issues here, and I'm not sure how this is going to come down. But yeah, this is kind of a big deal.
It's not a lot. It's 30.
It's 30 people out of the whole Cell Press enterprise, which, yeah, that's not a huge proportion, but the prominence of the University of California system within the scientific publishing world is out of proportion to their numbers.
Includes Jennifer Doudna and Liz Blackburn. So the Cell Press says, we're very sad and we hope to continue our relations in the future. Well, it's a good signal. I wish more people would do it. Unfortunately, people continue to publish in these journals, and they always will. I learned, by the way, by reading a book by an economist that in some institutions in China, if you publish in one of these Cell journals, you can get a 30% bonus in your salary. 30% of your salary bonus.
In one of these specific journals?
Yeah, like Cell or
Cell Science
Nature. So with that kind of incentive, they will always publish there, so it will never end, right?
Until there are more publications that they can publish in that are...
Well, they don't have the cachet, that's the problem.
And as long as hiring committees look down a stack of CVs and say, well, do they have any Cell Science or Nature papers? No, then we'll pass on them. That is going to drive people to publish in those journals.
It's a self-feeding machine.
Yeah.
Self-perpetuating.
It is self-feeding. I don't know how to deal with it. I saw on Twitter the other day, someone said, hey, there's a myth that you need a Cell Science Nature paper to get a job, but I got one at Berkeley, and I felt like answering, it's still a myth. You just happen to be an exception.
It's still, just the exception does not disprove.
Yeah. Well, unfortunate. I guess we could have some version of a social media outcry and public shaming, but that might be a step too far.
Usually doesn't accomplish anything.
Yeah, true. Momentary passings. Not usually enough.
Alan, can you take the last one from Anthony?
Sure. Anthony writes, virology news not far from home. I've found that raccoons here in Jersey City can at times be very bold without appearing to be ill. So the old rule of wildlife not to be scared of people as a sign of rabies is no longer true. Sick animals become harder to detect. And this is a little news article. 10 raccoons test positive for rabies in New Jersey town. Cops had to shoot one after it charged at a man walking his dog.
So, you used to say that if a raccoon was behaving badly, it probably had rabies. And he's saying that in general, raccoons are behaving badly without rabies. So.
Just raccoons. Yeah. I know very few animals. I go to Central Park every single day. I knew very few animals that will wander away from us. I've had squirrels try to charge me.
Really?
Yeah.
Yeah, urban animals, but raccoons have been behaving badly as long as there have been raccoons. This is just they steal trash. They become accustomed to humans. They're bold. The key is if they're doing stuff that's really weird, like charging at somebody who's walking a dog, that's a sick animal. And if you see them out during the day, they're nocturnal. That's probably an animal that's not in good shape.
Yeah.
But just, you go out and tell them to get out of your trash can and they look at you like, yeah, what are you going to do about it? That's normal raccoon behavior.
They're bold from...
But they also are huge reservoirs for rabies, so
not great.
It may just be safe to assume that if you see a raccoon, it's probably a rabbit.
Although, try not to ever pet a raccoon. Yeah, I think that's one of the morals of this is never try to pet them.
You shouldn't pet a growling dog.
Don't interact with wild animals like that.
My neighbor had a coyote in her backyard this week.
We have 2 in Central Park.
Coyotes?
2 coyotes. We had one for a while,
really?
And then
is it a
recently
couple?
This week, a second coyote has come up. They're BNT people. They're bridge and tunnel people.
They come through from the suburbs, yeah?
So yeah, apparently in the middle of the night, sometimes you'll have a coyote walk the tunnels, get lost in Central Park, and then decide that with the luscious amount of animals, it might as well make a home.
It's very interesting.
It plays with the dogs.
Is it male and female?
I'm not sure. I've not seen the second one. I only ever saw the first
one. So
they
may reproduce and make a quasi-species.
Or we could get, as I was talking before, the koi dogs. So there's the mixing between the dogs and coyotes that's been happening in some places. Yeah.
All right.
What is a species?
Let's do some picks. Alan, what do you have for us?
I have another one of these photography award things, and this is the Astronomy Photographer of the Year award. And these are just gorgeous to look at, and I like really cool astronomy photos and looking up at the stars and just kind of reminding ourselves of our place in the universe. And I think these are some stunning images that also, in some cases, illustrate some really cool stuff.
Hey, there's an aurora here.
Yes.
I was talking about it.
And there's the edge of the sun, which has a corona.
See, isn't that cool?
That's beautiful.
Can't see it on your phone.
Oh, kids these
days.
You
don't have the show notes. That's right. Sorry. I didn't send you the show notes.
I was not sending the beautiful photo on the show notes. That might become a phone background.
You have a Gmail account?
Yeah.
Let me send it to you.
This is awesome. We saw the Northern Lights when we were on our rapid.
Just emailed you a link.
I saw the Northern Lights once. I went to Iceland for a couple of days with one of my friends, and we walked around, we drove around, we couldn't find them anywhere. We take a flight out of Iceland back to England. The pilot goes, "Everyone stay calm, but if you look to the left, there you can see the Northern Lights." The entire plane got up, ran to the windows. We tilted partly down.
No, you didn't tilt.
We did. I think that probably might have been teasing us, but
yeah.
And it was this beautiful. We were over the clouds, and these green lights were just dancing above them. It was gorgeous.
I like this one of the sun, which is very cool.
Yes,
Alan. Speaking of planes, I flew back from Denmark on SAS.
Okay.
What a great airline.
Oh,
great people. Really nice. And I'm used to flying United, okay? The pilot was great. So, on an American plane, it bumps a little, they turn the seatbelt sign on. This guy didn't turn it on once. It's like, bump, oh, what's a little bump? What's a little bump?
Be a little resilient.
It was just a beautiful, just wonderful flying. And then when we were going to land, he said, okay, he was Danish. The runway at Newark is a little bit short, so we're going to brake more than usual. And I didn't notice anything because I'm used to Newark. But it was just such a good airline. And then someone said, most international airlines are better than United.
Yeah. Also, Norwegian is an amazing airline to fly.
I've heard that too, yeah.
You can, pretty cheap tickets. I now sound like I'm talking industry, but
they're nice. The people were nice.
Yeah.
That's the thing. I'm just used to being scowled at and, spilling water on me. They were just nice. So if you have a chance, fly SAS. Really cool.
Or Norwegian. Still a big fan.
Rich, what do you have?
I just realized, Alan, that these pictures are nested, right? If you click on a link from a given picture, you can get a whole bunch of other pictures that were submitted as well. You could, these are amazing.
Yeah, you can look at the short list of photos in each category.
They're nested like coronaviruses.
There you go.
Did you get the link?
Yeah, I am now looking. I've joined the technological era.
Rich, what do you have?
Just a little something that caught my attention. This is in the phys.org publication titled "Urban Living Leads to High Cholesterol in Crows."
I love it.
From the American Ornithological Society. And I did not try to go to the primary literature here. Just go for it, okay? And it
I love the illustration at the top with the crow pulling a burger out of a fast food wrapper.
Wait, which fast food?
Basically, this summarizes
It's McDonald's. I can see the art.
Maybe it's the Impossible Burger.
Yeah.
This summarizes an article where some investigators sampled cholesterol in both urban and rural crows and found that the urban crows had high cholesterol, which is because they're eating junk. They even did a control where they took some rural crows and fed them a bunch of cheeseburgers, McDonald's.
Yeah, right? That's right. They fed them.
And their cholesterol went up, which I thought was pretty cool.
So if they'd used Impossible Burgers, it would have been fine, right?
As it turns out,
the cholesterol of the Impossible Burger.
A more difficult experiment to do is to figure out whether there's any consequence of the high cholesterol in the crows, which they couldn't really figure out. They apparently urban crows live longer than rural crows anyway, probably because they got more food, even if it is high in cholesterol. So, but at any rate, I just thought that was cool.
If Dixon were here, he would remind us that the crows were the first to die when West Nile virus came to New York City.
Really? Why?
Because they had never encountered the virus before, and they got infected and died.
The virus could survive in crows.
Yeah, well, it grows in other birds. That's how it's transmitted. The mosquito-bird cycle is the usual transmission cycle, and wild birds are used to it. But crows here were not. So
that is still
so we had a murder of murders.
Yes,
exactly. Very good.
It's still one of the coolest sort of studies on virus emergence is the pictures of West Nile sweeping across the U.S. Over time. It's amazing.
I would love that link.
It's a cool story. Did you get Dixon's book, West Nile Story?
Me? No.
How about you, Kiki?
No, where is this?
Oh, I'll get you a copy from his office out there. It's a little story of what happened, including the bird story.
Does he also have sharks and jets?
No, just birds. Do you have a pick?
Yeah, I found a couple, and then during this conversation found a new one, which I think I'd rather do, which is reversal of epigenetic aging and immunosenescence? I can't say that word.
Immunosenescence?
Yeah, I feel that. Yeah, immunosenescence trends in humans. It's saying that they fed a cocktail to a group of men around in their 50s that was able to slow down what they call or reverse their epigenetic
clock.
clock.
Sign up for a study where they feed me a cocktail?
Well, it says it took...
a cocktail of drugs, I see.
It said that it took about, on average, 2.5 years off of their epigenetic clock of life. So it made their epigenetics 2.5 years younger than would have been expected after a year on this cocktail. Which I find fascinating. And also interesting because I've never heard the term epigenetic clock before.
What journal is it in?
This is in Aging Cell.
Okay.
Not sure if it's I
guess Alan found it.
Yeah, it's the copies of Cell that are way back on the library shelves.
Yeah, this was September 8th.
No. It's a journal called Aging Cell. It's an open access paper apparently.
Cool. Yeah, it's Gregory Fahey, Robert Brooke, James Watson.
Not the James Watson.
No.
James P. Watson, Zinaida Good, Shreyas Vasanawala, Holden Maker, Michael Leopold, David Lynn, Michael Kober, and Steve Horvath.
Cool.
Yeah.
All right. My pick is a 1953 book written by Salvador Luria when he was a professor at the University of Illinois. It's called General Virology. It is free. You can download a copy at many links. I'll give you one. And I want you to look at this because these are the early days of virology when it's starting to become a quantitative science. And, he says that in the introduction, and it's really a nice thing to look at what we used to think about viruses and what we thought the issues were. And we don't know a lot about viruses at the time, but this book is based on a course that he wrote, and he tries to look at principles, not talk about individual viruses, right? And he says about how it's important if you want to learn a field to look for the concepts and the principles. And so this is actually what we based our textbook on when we first issued it many years ago, the idea that you do principles, and we actually got it from his book because he said that's the way to teach. And this is when virology is becoming a biological science, because up until then it wasn't clear what a virus was. Is it a chemical? Is it something else? The first guy to crystallize viruses, Wendell Stanley, thought they were infectious proteins because he purified tobacco mosaic virus and found 95% protein. And he said, viruses are infectious proteins. And, well, he ignored the 5% phosphate that he detected in it that happened to be part of the RNA.
As we all do. Talking about infectious proteins, though.
Yes, we now know they exist, but Wendell didn't.
True.
So anyway, General Virology, S.E. Luria, free download. Check it out. We have a couple of listener picks. Sindhu gave this to me while I was in Stockholm. The Swedish solar system, or I should say Sweden solar system. She's a PhD student with Anna Smed Sorensen, who was on my TWiV there. So in Sweden, they have markers for the planets. And it starts in Stockholm, where there's this big spherical building called the globe or the sun, and that's the sun. And then all the other ones are lined up throughout the country, according to scale. Right?
To scale.
Yes.
Nice.
And there's Mercury and Venus. So where is Mercury? It's 3 kilometers away from the sun.
Pluto is 300 kilometers away from the sun.
3 kilometers away from the sun. So
she gave me a printout here with all of the sites located on Sweden. You can go visit them all if you want.
This summer.
Yeah, I was looking for a link to a map of the whole.
I think it's on the website. I think it should be directions and map.
Oh, directions and
there you go. Yeah, if you go to the left, directions and map.
Oh, okay,
you'll see it.
cool.
It's there. The whole thing from top to bottom. Isn't that cool?
Yeah.
I think there's one in the U.S. that starts in Ithaca where Carl Sagan was. But I don't know.
I think there was, yeah. I'm not sure if it's still maintained.
So over at Cornell.
What's that?
Ithaca, Cornell.
Yeah, I went to Cornell.
Really?
Yeah, that's why I know this. But it wasn't there when I was a student because, well, it wasn't there, period. I don't know why. All right, Anthony gives us a video with Eugene Koonin, The Surprising World of Viruses with Eugene Koonin, and it's old, 2016.
It's not old.
You don't think so?
I
thought you would agree with me, actually.
It's a 3 years, no. That's old, then I'm now getting old.
And Paul writes to the Twivlers, originally cherry flavored. Do you know what a Twivler is?
Twizzler.
Yeah.
Yeah, but he made it
That just made me really excited. I'm a big fan of the Twizzlers.
I think you'll appreciate Malcolm Gladwell's take on the path to retroviruses, and he provides a link to episode 40 of Revisionist History, number 40, The Obscure Virus Club. It would be great to hear your thoughts on what he gets right and wrong. Okay, now, when this dropped a couple of weeks ago, everyone went gaga on Twitter. Oh my God, Malcolm is talking about viruses. Nobody goes gaga when we drop a TWiV, right? Okay, all that aside. That's, Malcolm Gladwell. He's got a nice voice. He does these highly produced podcasts with, lots of music, and he develops a narrative by having people talk. And so I listened to it yesterday on the way home because I wanted to answer Paul. So the Obscure Virus Club, what this is about is 3 people, Ludwig Gross, Howard Temin, and Robert Gallo. And it's all about retroviruses, human retroviruses, and the story to discovering them. And Ludwig Gross discovered murine leukemia viruses. Howard Temin figured out that retroviruses had reverse transcriptase in them and made DNA copies which integrated into genomes. Okay, so it's all good up to there. And he interviews David Baltimore a lot, which is really great because he says, I did the experiments in 2 days to discover reverse transcriptase, and that got him a Nobel Prize along with Temin. That's brilliant. David is always brilliant, but it's really good. Can't interview Temin, of course, because he's dead, but he talks to his wife and daughter. It's very light on science, but it's mostly a social thing, and that's what Gladwell does really well. Let me tell you what I think about it. 2 things. First of all, in the very beginning, he talks about Rouse at Rockefeller discovering that a tumor of chickens can be passed to another chicken. Malcolm says, he took the tumor, he ground it up, and he injected it into another chicken, and it caused the tumor. And what's missing from that? The most important part of the experiment, he filtered it first. That's why he could say it was a virus.
Because of the size.
Exactly. All right. So I object to him leaving that out, but Malcolm would say, oh, it doesn't matter. Okay. Second problem, and there are just three I will go through. When Temin first had this idea that retroviruses had an enzyme that made DNA from RNA, reversed the central dogma, he called Francis Crick. And Gladwell says, Crick of Watson and Crick fame, the discoverers of DNA. And that bugged me, too, because now all his listeners, and there are tens of hundreds of thousands of listeners, think that they discovered DNA. Oh, no. And again, Gladwell will say, well, it doesn't matter. But the most important problem with this is Robert Gallo. And he interviewed him extensively because he's alive. Now, Gallo did one very important thing in his career. He figured out how to grow T cells in culture, right? You give them IL-2. And he used that to identify the first human retrovirus, HTLV1, from people with leukemia. This is undoubtedly an important discovery. However, Gladwell then goes on to give him credit for discovering HIV. Oh. And that isn't correct.
So who did discover HIV for your listeners?
Montagnier and Barré-Sinoussi at the Pasteur in France. Gallo got a sample from them and pulled the same virus out that they had already pulled out. It was a contaminant, so he didn't get it. And that's why he didn't get the Nobel Prize. They did. So I don't think having Gallo in there was a good idea. I would have done it totally differently. But I like Ludwig Gross. I like Rouse. I should have spent more time on Rouse, I think. Should have given David Baltimore way more time. I would have made it Gross, Temin, Baltimore. So that's my thought. Do you guys, anybody hear it? You haven't heard it, Kiki?
I just started listening to it. I have it on my list here.
So listen to it. I think it's worth it. I'm
not a big Malcolm Gladwell fan.
Yeah, I mean, you have to like him.
He definitely does not let the facts get in the way of a good story.
Exactly. Yeah. Very well put. There you go. He does a highly produced podcast, which we don't do. And one last thing. He thanks his team at the end. His team who do all the footwork for him, right?
They do all of his makeup for the podcast.
Well, right. They go do his research. Oh, you should interview this person. You should interview this person. At one point, Malcolm says, I found the speech that Gallo read at Temin's obituary. You didn't find anything. Your assistant found it for you. We don't have any team.
Author.
We have us here on TWiV and all the other podcasts. Anyway.
Oh, and the readers, listeners who write in.
Yeah,
it's a community.
We have a good community here.
It's a great community, I'd say.
That is TWiV number 566. You can find all of the notes and links at microbe.tv/TWiV. If you listen, you do so on a phone or tablet. Please subscribe. A lot of people just do one-offs. I don't think you should do that because if you subscribe, you get every episode, and you never know when there's going to be one that appeals to you. And we know how many people listen. That's important to us. So please subscribe. Do you subscribe?
I do subscribe. It's so much easier. I subscribe to only about three podcasts, and this shows up so that I don't forget that it's happened, and I don't have to keep researching every time.
There you go. That's a perfect reason. And then we see we got how many subscribers, and that's important to us for raising money. Speaking of money, if you like what we do, go over to microbe.tv/contribute. You can give us $1 a month using Patreon or PayPal, and that helps with our expenses and it would allow us to travel more. I would love to take more people with me when I go on these trips, and I can't all the time, so we'd like to be able to do that. And questions and comments, go to TWiV at microbe.tv. Today we've had a compact TWiV in terms of hosts.
Still 2 hours long.
Yes, Alan's always quick to point that out.
We're the virus of hosts right now.
You didn't have a bad time, right, Alan?
No, it was not objecting at all.
It was
just kind of preempting people writing it. That was not a compact TWiV.
Compact in terms of host. Rich Condit is a emeritus professor, University of Florida, Gainesville. He's currently residing in Austin, Texas. Thank you, Rich.
Sure enough. Always a good time.
Alan Dove's at turbidplaque.com. He's on Twitter as Alan Dove. Thanks, Alan.
Thank you. It's always a pleasure.
Here in studio is Kiki Warren, who is a student at Columbia University. You may wonder what her connection is to TWiV. Well, she took my virology course, right?
And I'm an avid reader and person of interest in virology.
In general, yeah?
Yeah, the person of interest sounds a little bit on the other side.
Yeah.
Thanks
I'm
for coming.
the top list.
Thanks for coming. Study hard.
You can follow me at Kiki Warren on Instagram if you would like to see dog photos.
Kiki Warren. All right, thanks for coming. I'm Vincent Racaniello. You can find me at virology.ws. Thanks to ASV and ASM for their support of TWiV and Ronald Jenkins for the music. This episode of TWiV was recorded, edited, and posted by Vincent Racaniello. I say that because Malcolm Gladwell says, here's my team. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.